Welcome to our channel Epistem, and today the topic of our discussion is master mix composition in PCR and the role of each component. So what is the master mix? Now master mix is actually a mixture of all the components, enzymatic components and the optimization components that are required for a PCR process to run. So it actually contains the DNA polymerase, primer, template, MgCl2, DNTPs, nuclease-free water and some other components which we will discuss later in the video. The importance of the master mix is that it eliminates the need of individually adding the precise amount of the enzymes, buffers, cofactors, etc. and thus it saves the time and the effort of the researchers. Now let's talk about the components of master mix. First one is DNTPs. DNTP stands for deoxynucleoside triphosphate. So we have 4 DNTPs: DATP, DGTP, DTTP, DCTP. Now these are the building blocks that are required for the growing strand to elongate. Now we see in this diagram, the blue strand we see, it is the template strand and the yellow one is the primer. Now the primer has 3' end, and from the 3' end the DNTPs are added one by one complementary to the template strand, and these DNTPs are added by the DNA polymerase. The next one is primer. So we need 2 primers in our PCR because you see the DNA molecule that we use, they have 2 strands, so each strand requires 1 primer. So here in this diagram, the primer that is binding to the 5' to 3' strand is called the forward primer, and the primer that is binding to the 3' to 5' strand is called the reverse primer. These primers are usually prepared in labs according to our need, and they usually have a high content of GC in the strand. It is because when we see these bases, guanine and cytosine, we see that 3 hydrogen bonds are formed in between them. So due to the presence of high number of hydrogen bondings, this create a stable structure of the primer and the template complex. So this can't be achieved if we have AT, because A and T, they have only 2 hydrogen bonds in between them. So we prefer having high guanine and cytosine concentrated primer because they will increase the stability of the primer and the template complex. Next we have the Taq DNA polymerase. Now for the PCR process, we usually prefer the Taq DNA polymerase because PCR occurs at a very high temperature of 95 degrees centigrade. Now if we use the DNA polymerase of that is from E. coli, then these bacteria, the DNA polymerase which they produce, they are not thermally stable, so they denature at a high temperature. So that's why we use the Thermus aquaticus DNA polymerase, because this bacteria is found in the hot springs and their enzyme has an added capability to survive at a high temperature. So the function, now we will discuss how it actually works. So this is the DNA template strands. Along with that, the primers are also added. Now you see the 3' ends, that is the 3' OH of these primers are free. So the Taq DNA polymerase will identify these ends and they will bind to it, and from this 3' end the DNTPs are added and the strand is elongated. So that as I already mentioned, that the DNA polymerase from E. coli that has been earlier used, now it is not used because they denature at a high temperature. And all polymerase, they synthesize the DNA from 5' to 3' directions. The next component is buffer. Now a pH level of 8 to 8.3 is essential for the high efficiency of DNA polymerase, stacked DNA polymerase to function. The next is the K plus ion, which we obtain from the KCl. Now this K plus ion actually, they help in primer annealing, that is primer binding to the template strand. So you see, this is our DNA and this is our primer. So the DNA, we have the DNA backbone has negative charge on them. It is because the backbone of the DNA has the phosphate group, which provides a negative charge to our DNA molecule. And we have the primer. The primer is also negatively charged. Now when the negatively charged meet, they cause electrostatic repulsion, which you all know. So the binding is not efficient. To prevent this problem, we add these K plus ions in the backbone of our DNA molecule. Now see, now these negative charge are masked and the primer can easily bind. The K plus ion has also role in the prevention of degradation of our DNA molecule. The next component is MgCl2. Now MgCl2, they act as a cofactor for our DNA polymerase enzyme. So to understand this concept, let's refer to this picture. In this picture, we have the DNA polymerase in the left hand side, and the blue strand is the template strand and the red colored strand is the primer. Now there are two Mg2 plus ions here. So the topmost Mg2 plus ion, they coordinates with the 3' OH group, the phosphate group and the aspartate that are present inside the DNA polymerase. And the bottom Mg2 plus ion, they coordinate with the pyrophosphate. Pyrophosphate means the beta and the gamma phosphate groups and the two aspartates that are present inside the DNA polymerase. Now the function of Mg2 plus ion is that it facilitates the attack of the 3' OH to the alpha phosphate group. And the role of Mg2 plus ion that is present at the bottom is that it facilitates the displacement of the pyrophosphate. And as a result of this, the incoming DNTP gets added to the 3' OH group of the primer. And thus, this way the strand elongation is possible. So, this Mg2 plus ion act as a cofactor for our DNA polymerase enzyme. And in one line, if you want to say the role of the Mg2 plus ion, then it helps in the coordination of the interaction of our 3' OH end of primer and the phosphate group of that incoming DNTP, that is denoted here in the black. So in the phosphate group of the incoming DNTP and helps in the DNA polymerization. The next component is the mineral oil. So mineral oil is a optional component because if we use a PCR machine, now machine has a lid on it, so there is no risk of evaporation of the sample, because as I already mentioned earlier that the PCR takes place at a very high temperature, and so there are chances that our sample may get evaporated due to this heating. And so if we use a PCR machine, we don't use the mineral oil, but other than that we use the mineral oil to prevent the evaporation. Next we need nuclease free water, because if we have nuclease in our water, these nuclease will degrade our nucleic acid. So it will only defeat our cause of the PCR process. So make sure the water you use have no nuclease in it. And lastly, we need the template strand, which is very obvious for this process. Thank you for watching our video.